At the molecular level, an active pharmaceutical ingredient is a symmetrical, pristine structure engineered to perform a specific biological function. But during the synthesis process, secondary chemical structures can unexpectedly form and attach themselves to that primary molecule. These are related substances. They are impurities that share a chemical link to the main drug, appearing either as side products during manufacturing or as the result of the drug molecule breaking down over time. Even in small concentrations, these variables can change the way a drug behaves. They have the potential to impact the medication's shelf life and how effectively the body processes the active ingredient. To manage these risks, laboratories use this framework to identify specific chemical impurities. First are process-related impurities, unreacted materials or byproducts from synthesis. Second are degradation products, occurring when a drug is exposed to oxidation or moisture over time. Third are residual organic solvents remaining after purification. Fourth are inorganic impurities, like leftover catalysts. Fifth are elemental impurities, focusing on trace metals. Finally, polymorphic forms are different crystalline arrangements that can change how a pill dissolves. Because these contaminants range from volatile solvents to heavy metals, no single test can identify them all. Impurity risks exist at every stage of production, originating from raw materials or incomplete chemical conversions during manufacturing. Post-production, environmental variables like light and heat can trigger new products, while packaging materials can leach compounds into the medicine. To identify a specific substance within this life cycle, analysts require a reference standard. This is a pure sample of the impurity that acts as a known baseline for comparison during testing. Because these variables appear from the initial chemical vat to the patient's cabinet, quality control functions as a continuous validation loop. Testing protocols utilize specialized machinery designed to detect the unique physical properties of each impurity class. In an HPLC readout, the machine separates the active ingredient from other substances, plotting distinct peaks to precisely measure organic impurities. Gas chromatography then identifies volatile components, vaporizing the sample to measure solvent residues like methanol or acetone. For elemental impurities like lead or arsenic, labs use inductively coupled plasma mass spectrometry, which breaks the sample down to its elemental components for measurement. UV and infrared spectroscopy are also utilized for preliminary screening, using light absorption to confirm the chemical structure of unknown substances. These technologies convert invisible chemical variations into the quantitative data required for pharmaceutical analysis. Once an impurity is measured, analysts refer to a standardized framework to determine if that specific concentration is acceptable for clinical use. The International Council for Harmonization, or ICH, provides these global standards. Their Q3 guidelines set specific limits for drug substances, products, solvents, and elemental impurities. These limits are calculated based on daily patient exposure data. They provide a realistic threshold where a drug can be manufactured consistently while maintaining a high safety profile. These guidelines translate complex data into the operational parameters used to approve a batch for the market. The identification and control of related substances is a primary indicator of a drug's stability and chemical consistency. Adhering to these analytical pathways is a core requirement of good manufacturing practice and ensures that every dose performs as intended. To learn more about the technical standards of pharmaceutical quality control, subscribe to the channel. Staying current with these testing methodologies is essential for navigating the evolving requirements of pharmaceutical safety.